Hello everyone, this is Geethanjali. In today's video, we'll be learning about translation. So, translation is nothing but protein synthesis. Translation is the process of polymerization of amino acids to form a polypeptide chain, which is basically a protein. So, we have learned that ribosomes help in protein synthesis, and these are present both in eukaryotes and prokaryotes. And this protein synthesis which is there, it takes place both in eukaryotes and prokaryotes in the cytoplasm, basically where the ribosomes are present. So, to start off with translation, we need the tRNA molecule. So, the tRNA molecule which is there, it first has to get charged. What is the meaning of charged? So, charge is nothing but amino acetylation of the tRNA. That means, and its respective amino acid gets linked to the tRNA at its 3' end, as we learned while studying about the tRNA. So this happens in the presence of ATP, as every metabolic process requires ATP. So here also the attachment of the amino acid to the 3' end will require ATP, and that is why we also call this the charging of tRNA. So we know that the initiator codon in the mRNA will always be AUG. So corresponding to that, the amino acid having methionine, the tRNA carrying methionine, which is called, this tRNA is called the initiator tRNA. So this initiate tRNA is ready. So first what happens is that we have our mRNA over here. So this process is called initiation. Okay, first the charging of the tRNA takes place, and then to the mRNA that we have, the lower subunit of ribosome gets attached. So that is when the process gets initiated. So the ribosome basically has two sites. Okay, it has a P site and a site. First, the initiator tRNA comes and sits in the P site. Okay, it fits in the P site. So when it fits in the P site, our larger subunit, that is the 50S subunit, in case of prokaryotes. Okay, in case of eukaryotes, it would have been 40S, and here it would have been 60S. So they come and it sits on top of our lower subunit, such that the P sites are P and A sites are perfectly joined. Okay, so this is basically our initiation process in the protein synthesis. So in initiation process, the charging of DNA happens, and then the lower subunit first comes and attached to the mRNA, then the tRNA comes, and then again our larger subunit also enters. Now, after that, the reading the next codon. So here we have UUU. So another tRNA, okay, which is responsible for carrying phenylalanine, because this is UUU, so this codes for phenylalanine. So the tRNA carrying phenylalanine comes and attaches itself here to the A site. So here this was P site previously, and our tRNA carrying methionine has been placed here. So next our tRNA carrying phenylalanine will come and sit in this A site. So when this happens, our two amino acids are in very close approximation. Okay, they're very close to each other. So because of this and another enzyme which we call as ribozyme or the 23S RNA, this is called the peptide transferase. So this enzyme catalyzes the formation of a peptide bond between the amino acids. So in case of eukaryotes, it will be 28S RNA, whereas in case of prokaryotes, it will be 23S RNA. So this 23S RNA is nothing but a ribozyme, and this basically forms the peptide bond between the amino acids when they are in this close proximity. So here, when the bond is formed, what happens is that this amino acid which was in the P site moves away, and this entire structure, this entire ribosome moves forward. So when this moves forward, when this entire thing moves forward, the P site comes to the place where the previous tRNA was, this UU tRNA was, and you will get something like this. So here the bond is formed and the methionine is on top of the phenylalanine like this. Okay. Then, as I told you, the ribosome moves, so this entire structure moves and goes forward. Then, whichever next codon is present in the A site, so this comes to the P site and here the A site. So let us say it was GGG. So the next tRNA carrying whichever amino acid the GGG is coding for, it comes and binds to the A site. So again the same thing happens. The ribozyme comes and it catalyzes the formation of a peptide bond between the amino acids because again they are in close proximity. So this keeps happening until ultimately we find a stop codon. So we know that there is no tRNA for the stop codon. So no stop codon comes, but there is something called the release factor which helps in the termination of this translation process. So this release factor comes and terminates this process of translation and also releases our polypeptide. So this will be our termination. So one thing here is that we have some parts of the mRNA which are not translated. Okay, these parts, suppose we have AUG over here, some code, and then UAA, and there must be some other nucleotides here too. So this region, okay, this region is called untranslated regions or UTR, untranslated region or UTRs. So UTRs, as I told you here, they are present both at the 5' end and at the 3' end. So they come before the 5' end and they also come after the 3' end. So these regions are untranslated, as you know. So they are known. Why do we need this basically? So they are known to be required for an efficient translation process. So for translation to go on smoothly and efficiently, they are known to be required. This was about translation.